So just first of all, my name is Danielle Bourque. I'm happy to be here. I actually was at the first PISCeS iteration 5 years ago. Holy crap. 5 years ago. I was a grad student 5 years ago. I didn't even have my data set completed. So I transitioned out of being a graduate student for Bob into consulting with SLR. And I'd like to talk about today one of my first industry case studies, I would say. So multiple lines of evidence were required to confirm SAR presence at a wetland remediation site. So I'm going to tell you a little bit about my site. And due to the nature of this project, I can't disclose the client or the exact location, but they did give me permission to talk about this work in a certain way. So bear with me. I can tell you that the site was located in the Fraser Valley of British Columbia, Canada. And previously, this site was used for agricultural purposes as well as an illegal landfill, so not nice. Currently, and since 2014 now, it has become a wetland remediation project, so trying to restore riparian and wetland habitat. And on this site, during this project, it was observed, or it was hoped, that with remediation efforts, we could promote the presence of a few key species at risk. One of them being Oregon spotted frog, which is endangered in British Columbia, northern red-legged frog, which is currently special concern, and Pacific water shrew, which is a semi-aquatic mammal, and that is endangered. In terms of invasives, so in my work, I was not personally concerned in my component of the project about Himalayan blackberry or canary reedgrass, but I did want to mention that as a part of this work, there was ongoing management efforts to remove those two from the site. But what my concern as the molecular ecologist was about was the green frog and American bullfrog, both which present problems for food and habitat for the endangered Oregon spotted frog. They're a competitor. Oh, and I put a little circle. There was also the construction of a bat condo on site to promote bat presence. And this bottom photo is just another representation of what the habitat looks like presently. Okay, no problem. I just like to walk around, so. Oh, did it go twice? Yes, it did. So, sorry, I'm getting used to the keyboard. So our objectives were, we wanted to use molecular methods, so eDNA, to identify site use by species at risk and other wildlife to support the protection of SAR critical habitat. We also wanted to identify threats to SAR, so the presence of some of these invasive amphibian species, and evaluate seasonal use of the site by species at risk. And as you can see from the image, you can already tell what the equipment I used to do my sampling was. So if you're not familiar, that blue backpack is made by Smith Root. It was previously called the Andy, but is now called the Environmental DNA Sampler. So that was the equipment I used. And I'm going to go over my methodology. So I got to go to this site 2 times in 2021. The first time was in February, so before the spawning period for these amphibians. And I collected 24 field samples as well as 2 field control samples, one for the 2 days that I was on the site. And each of my field samples, at this project, my target volume was always 5 liters, but if a filter clogs before then that's what you get. So that's what the around 5 means. So per filter, 5. And I want to mention that these field sample collections are all spatially explicit. I don't take technical replicates at each of my stations. I maximize, with the budget I have, spatial diversity in my sampling. And I can't show you a picture of the site, so you'll have to take my word for it, that each of these samples were about 20 to 50 meters apart. And in terms of coverage of these wetland areas, I covered them quite well with that range. They weren't very big areas. And due to funds, we sampled for the next step, for actual analyses, we sampled a subset of those 24, so down to 16 in the first visit. And we analyzed these for eDNA for 2 ways. The first was qPCR using Bureau Veritas laboratories. So they have targeted assays for the northern red-legged frog, Oregon spotted frog, American bullfrog, and Pacific water shrew, and I've listed the target regions for those assays there. We also conducted 12S metabarcoding, so we were using the Batra and My Mammal publicly available assays to do that. And we subcontract the metabarcoding to Lab Services Division at the University of Guelph. I got to return in November and conduct a second visit. And on this program, we only used metabarcoding, and that was a funding decision and based on what we got from our first visit. I also want to point out that November is also not the spawning time, but sometimes the time you get to go to site is not always when you want to go to site. But I know some of you can relate to that problem. Oh, we're working again. So, on my first visit in February, interesting conditions. There was still some ice present on the water. And if you're going for water samples, you need to work around ice. And it was quite ironic that as you're there, you go, hey, what's that? That's a frog. And you're not detecting a frog sitting on ice in a water sample. But visual observations still count. So that's a green frog. So, it's always nice to see a frog present in a wetland, but it would have been nice to see one of the species we're trying to promote and not the exotic species competing with our species at risk. But still a sighting. And here's what we actually found in terms of eDNA. So, again, remember on this trip there was quantitative PCR as well as metabarcoding happening in tandem. And in terms of detections, for northern red-legged frog, we were able to detect it in 1 out of 8 technical reps using the quantitative PCR assay. No other detections were made for that species. That green frog photo I showed you, he was spotted visually. That was not an eDNA detection, but that still counts. And we had no detections in either method for Oregon spotted frog, American bullfrog, and I'm going to get more into it more later about the Pacific water shrew, as we had 10 instances or detections of that. And I should clarify that one instance is from one of my samples. So there were 10 samples that had a hit for the genus that Pacific water shrew belongs to. And if I had to postulate why we had sort of low detection to success in general with eDNA, I do think there is a seasonal component to this. There could be low target organism activity because of the time of year, and that could translate to low available DNA. And of course, the restrictions of working around the ice patches. In November, things got a little more exciting. So if you're not familiar, the frog on the left, that is an Oregon spotted frog, and that is one of our photos. And we also were lucky to encounter a northwestern salamander on the site. So maybe you're catching on to the theme here that even when you're out doing an environmental DNA survey, either just visual encounters and any other information you're gathering while you're there is all valuable towards your objective. Oh my gosh, and as you remember in November in DC, they had a very serious flooding event, and we beat that by a week. If that flooding event had occurred before my scheduled date, we absolutely would have had to move the survey. That inundation of flooding would have been a huge problem for the wetland and potentially diluting target DNA from anything that was there. Massive events like that need to be worked around in your field programs. So if we look at what was found this trip, this time, and again, only metabarcoding now for this trip. So no detection of northern red-legged frog. However, we did have 2 samples light up for a green frog and Oregon spotted frog. This is very interesting. So one sample had a match, 98%, to Columbia spotted frog. And that was interesting because the experts on my team said, well, you're not in the range of Columbia spotted frog at your site. So what was interesting about this particular sample, and again, very important to have good field notes and for photographs. So if we go back, that frog. So what happened was, I get a text from my team who's on site doing the vegetation component of the program. They go, Danielle, there's a frog right here. Come over here. So I'm with the backpack. I run over. And I stick the wand, the sample, right behind this frog, like as close as I can get before he leaps away. And we get good photos of the frog while I do it. And because of this encounter that happened concurrently to the sampling, we have good confidence that that frog we took a photo of was the source of DNA in the sample. And an internal expert on our team looked at that photo of the frog and said, yes, you have Oregon spotted frog. So this is very interesting, because if we only had metabarcoding, I would have to go to the client and explain, okay, with the database matches, we're seeing Columbia spotted frog, but because of known ranges, we can assume it's something else. But in terms of client confidence, that added visual encounter was key in this case to demonstrate site use by a SAR amphibian, and points to the need for more sequence data for our species at risk, for common metabarcoding markers like 12S. Now I want to get into more about this Pacific water shrew. What was happening was with the qPCR tests in the first visit in February, we had no detections. Maybe Pacific water shrew was in there. But as many of us know, it's very difficult to prove absence with eDNA. And that was not within the scope of this study. So when I went back in with metabarcoding, my mammal, I could only get matches up to 97% to Sorex, which is the genus the Pacific water shrew belongs in. But that's also the same genus as common water shrew and common shrew. So I was unable to conclude whether Pacific water shrew, the species at risk, is utilizing this site. And again, we did spot a northwestern salamander, just physically. We didn't actually detect it in the watercourse, but it wasn't found in the watercourse. So perhaps at the time of my sampling, it just wasn't in the water recently. And we did see, unfortunately, sometimes frogs get away. They see you coming, you don't jump in, and you're not sure what it was. And I wasn't around at that time. So in conclusion, both qPCR and metabarcoding techniques identified seasonal use, because we were there 2 different times a year, by species at risk to support the protection of SAR critical habitat. We also identified presence of a competitor to Oregon spotted frog, that's the green frog. And that was visually, confirmation from an expert, and environmental DNA. And we couldn't not confirm the presence or absence of Pacific water shrew. Further testing or methods, perhaps trapping, but that requires permits and more funding, would be required to investigate further Pacific water shrew on the site. And as I mentioned before, there's a continued need for publicly available species at risk sequence data for common metabarcoding markers like 12S. And of course, visual ID and knowledge of the species ecology is critical to success. I'd like to acknowledge my team at SLR, my field crew, my supervisors and reviewers, as well as I have a collaboration with the Hanner Laboratory that allows us to do these sorts of projects. And I'd like to know if you have any questions.